Aloha guys, Dr. Thomas coming at you once again. We're going to continue with our fascinating discussions, and today it's going to be about the nervous system. Not the easiest topic, but we'll do it. I've got confidence in you. Okay, there's two major parts of the nervous system, the central nervous system and the peripheral nervous system. The central nervous system is abbreviated CNS. You'll always hear that. Peripheral's PNS. How can you tell the difference? It's very simple. Sorry, there are airplanes and helicopters and all kinds of going overhead over there. The central nervous system is the brain and spinal cord. Everything else is the PNS. The central nervous system is the brain and spinal cord. Everything else is the PNS. Piece of cake. Functions of the nervous system. Sensory, in other words, pain, temperature, all the sensory information. Integrative, we're thinking about things. And motor. Motor makes things move. Remember the motor unit with the muscles. Cells that make up the nervous system. There's two broad types of cells. The functioning nerve cells are called neurons. We've talked about those before. They're the ones that actually generate the nerve signals and transmit the nerve signals. But there also are a number of neuroglia or glial cells. Now this says most abundant type, but I was preparing a video for my YouTube channel and the scientific work that I conduct, and it said that there were a lot more neurons than neuroglia. So, but those numbers change, guys. Science changes a lot. You know, that's another topic for another day. What are the neuroglia and glial cells? They support, protect, insulate, nourish, and generally care for the neurons. I hate it when I do that, but I do it a lot. Okay, here are some drawings of neuroglia. Astrocytes, ependymal cells, microglia, Schwann cells, oligodendrocytes. These are all the major types of neuroglia. Neuroglia do not generate nerve cells or conduct nerve cells. They support the neurons which do. Okay, we touched on this during tissues. Parts of a neuron: cell body, dendrites which bring information into the cell, and the axon which conducts it out of the cell. Bundles of axons are what is commonly known as nerves. Okay, and then axons have terminals, so other nerves attach to these dendrites and send in information. The average neuron has upwards of 40,000 connections. Oh yeah, you're going to love all this, guys. There's the same, depending on what source you believe, there's the same number of neurons in the brain as there are stars in the Milky Way galaxy. I always love that. Okay, and then these axon terminals junction with other nerves, or in the case of muscle, the muscles, you know, so it's really, really complicated. Okay, now the axon is of particular importance. Remember, axon is what we call nerves. Some of them have a myelin sheath around them. Here's the word. Now, the myelin is actually a fatty white yellowish color, so I don't know why they make it like turquoise here, but they did. And there are gaps in the myelin sheath called the nodes of Ranvier. What this does, guys, it lets the nerve signal jump between the gaps, so it speeds it up tremendously. Indeed, it does. Neural lemma is the nerve cell, the nerve membrane, axon terminal. Now, not all axons are myelinated, but many of them are. This is it, the myelin sheath. I think no, not the ependymal cells. One of the neuroglia produce the myelin. Types of neurons. They're sensory or afferent. Now, this is something you need to learn. Sensory information is going from the body up to the brain. Sensory information is going from the body up to the brain. It's called afferent with an A. Motor neurons are leaving the brain and going out to the muscles and glands to make them work. Motor neurons are leaving the brain and going out to the muscles and glands to make them work. They're called efferent with an E. Very important to know this. You might as well memorize it right now. Afferent goes up, efferent's coming down. Sensory information is afferent, motor information or signals are efferent. And then there are interneurons between them. White matter versus gray matter. It's very simple. White matter is myelinated with myelin sheaths, and gray matter is not. That's why I was fussing about the turquoise color because it's actually kind of white. White matter made up of myelinated axons. Why? It's faster. Conducts the electricity, as you were, because it's not electricity. It conducts the nerve signals faster. Gray matter is made up of unmyelinated axons, cell bodies and synapses. Synapses are the gaps between nerve. The nerve impulse, what is it? Electrical signals convey information along a neuron, also called an action potential, move along sensory or motor nerves. Now, it's very important because the way all this stuff makes it sound like it's electricity running through our nerves, just like the electricity runs through the wires in our wall. And that is not true. It is charged ions. Well, of course, all ions are charged. They wouldn't be an ion. It's ions moving in and out that creates the signal. Has an electrical quality, can be measured, but it's not electrical. It's not the movement of electrons through a wire. Okay, so it's very important to distinguish that, in my opinion. All right, now, this stuff can be pretty complex, guys, neurophysiology. But we're going to make it a little easier for you. Okay, here's the axon. Right at one of the nodes of Ranvier, they've taken and drawn a picture. So this is the neurolemma, the membrane around the cell, around the axon, that's permeable to ions. The two primary ions are sodium ions and potassium ions. Okay? During the resting phase, when there's no nerve signal, the neurolemma is said to be polarized. It's negative on the outside and positive on the inside. When it is stimulated, let's say we touch our finger to the hot stove and those pain signals are picked up by the body and end up in the brain. The thing that makes the nerve signal is a sudden inrush of sodium ions. So it depolarizes and changes it to a positive. And this signal is transmitted all the way down. That cascade effect of sodium ions start rushing in all the way along here. Of course, now remember with nodes of Ranvier, it's called saltatory conduction. It jumps between these knobs. So there's an electrical quality, but it's not electricity, it's ions. And then to repolarize, so it can fire again, it has to recharge itself. Potassium ions leak in, and it changes back and becomes negative again. Stimulation is also known as an action potential. So what causes the nerve signal? Well, it's already negative inside the nerve. And when you stimulate it, sodium ions rush in, make it positive, and that change is propagated down the axon and transmitted on whatever it's going to do. That is a nerve signal. It's Neurophysiology 101. Here it is, polarization. Potassium ions leak from the neuron, making it negative inside. Well, I can see they're positive. Determines resting membrane potential. When it's stimulated or depolarized, sodium ions rush in, which makes it positive. And then it has to repolarize, go back to where it can be stimulated again. And that occurs because potassium ions rush out. So it goes from negative to positive and then back to negative again. Okay? Hopefully you've got that. It's more complicated than that, but that's the basics of it right there, guys. The nerve impulse, what causes it to move? It's called an action potential. Forms at axon's beginning, regenerates along the axon's length, enters the axon terminal, releases acetylcholine from vesicles. Yeah, if it's a motor nerve, it does. There are many, many different kinds of neurotransmitters, guys. Acetylcholine is the one that's active in muscle movement. We talked about that. So here it is. See, it's at minus 90 is the resting potential. When it gets stimulated, it jumps way up to a plus 20, and then it repolarizes and gets ready. And during this time here, guys, the nerve cannot be stimulated again because it's recharging. It's like if you had a bow and arrow. You pull it back, and then it's polarized, and then when you release the arrow, boing, it depolarizes, and then you have to take and pull out another arrow and put it on there and notch it and get it ready. And then it's repolarized. Okay, here's the same drawings again. It's another node of Rondier. It's just showing, this time it's showing potassium and sodium. This is the reason, guys, that these electrolytes, which are what these are, have to be in a certain concentration in your body fluids or you'll die, because nerves will cease to function. So it's real important stuff. And then the signal is propagated, or it continues, all along down the axon. The sodium starts pouring in, and it makes a signal. It transmits the signal on down until it gets to the axon terminal. Okay. Myelin insulates the axon. It's very similar to insulation on wires. Very similar. Myelin exposes some axonal membrane at the nodes of Rondier. Action potentials jump quickly from node to node. Boing, boing, boing. They're comparing it to a kangaroo. How cute. What's the purpose of this? It speeds it up. It's much faster. It only has to depolarize at the gaps instead of all the way in between. Parts of the synapse. Now, the nervous system is ungodly, unimaginably complex. But where the axons come and they join with one of the, let's look at this picture here. Here's neuron A. Here's its axon. Here's neuron B. This is one of its dendrites. And the gap between these is called a synapse. Synaptic cleft. Receptors, neurotransmitters, and activators. So as the nerve impulse comes down, guys, these vesicles empty their neurotransmitters, their chemicals, and they migrate across the synaptic cleft or gap into certain receptor sites. Now again, this is using acetylcholine. So, and then we can't have it constantly being stimulated, or our muscles would go into tetany. That's exactly the way military nerve gases work, actually, some of them. So on the other side over here, it secretes acetylcholinesterase, which breaks it down. What amazes me, guys, is how we're able to make these incredibly fine movements and all with this. It's just mind-blowing. Nothing approaches it. Events of the synapse. Now again, this is a synapse that's using acetylcholine, but it's not the only one. Secreted from neuron A, diffuses across synaptic cleft, bound to receptors on neuron B. Neuron B is therefore activated and the signal is continued on. It might be modified a little bit. All right, we're doing good. Now, obviously, guys, I've covered a lot of material. Go back and look at it again. 4 major areas of the brain. Now, here's the brain. The face would be over here. Here's the spinal cord. There's the cerebrum. That's the main big hunk of the brain where higher thought takes place. The diencephalon, which we'll talk about later. The brain stem, which has 3 parts. And another little miniature type brain called the cerebellum. It's in the back. So this is the back of the head. The face would be out here. What's the 4 major areas of the brain? Cerebrum, diencephalon, brainstem, and cerebellum. Cerebrum, gray on the inside, white on the outside. Or gray on the outside, white on the inside. What makes it gray? Unmyelination. What makes it white? Myelinated fibers. There are many, many myelinated fibers in the body, but there are many unmyelinated also. The cerebral cortex is a thin layer of tissue of cells, neurons, on the outer portion of the cerebral hemispheres. And it's where all the higher thinking takes place. It's the highest of the highest. And it's made up of gray matter, so they even talk about, you need to use your gray matter. Outermost portion of the cerebrum. White matter makes up the bulk of the cerebrum, located right below the cerebral cortex. Now, why is it that way? Well, I mean, the short answer is that apparently the cerebral cortex doesn't need a great deal of speed, so it doesn't have to be myelinated. And I'm sure there are probably other reasons for that too. Markings. Now, every bulge on the brain has a name. They're convolutions. And every fissure or crack on the brain has a name, which are called sulci. These. And yes, Dr. Tom had to learn every single one of those. Every bulge has a name. Every crack has a name. Life is not fair, guys, when you start learning this stuff. It's a little easier than it looks, but it's horrendous. Okay, now we're running out of time. So we're going to pick up the next time. I'll see you then. Dr. Tom signing off.